Todd is an assistant professor at Colorado State University in molecular weed science. He completed his PhD at CSU, followed by postdocs at Western Australia and Germany. His research goal is to support sustainable weed management that helps contribute to sustainable cropping systems, including the molecular and genetic basis of complex herbicide resistance mechanisms, using next-generation sequencing to study complex traits in weeds, and developing molecular markers for rapid resistance diagnostics. Todd?
Thank you, Steve, for the introduction, and thanks everyone for joining in this webinar today. As you've seen, Pat has given you a range of fascinating examples by which weeds can evolve resistance to herbicides through target site mechanisms. In the second part of this webinar, we're going to talk about some non-target site resistance mechanisms and how those can even further complicate management of herbicide-resistant weeds. As Pat mentioned, we can classify resistance mechanisms broadly into target site category, which has been covered, and then the second category includes what we refer to as non-target site mechanisms, and these are essentially anything that is away from the site of action of the herbicide that prevents a full concentration of the active herbicide from reaching that target site. Different ways by which this can happen include metabolism of the herbicide. The plant is actually able to modify the herbicide into a different form that's no longer active, and we'll look at some examples of that. We also have several examples where the plant is able to reduce how much of the herbicide is taken up and translocates to the site of action. Keep in mind that while we do classify resistance mechanisms into these two categories, we know that really any trait which can slow down or prevent the herbicide from reaching its site of action or that reduces the toxic effect at the site of action can contribute to resistance. And it's certainly the case that these different mechanisms can combine within one individual to confer even greater levels of resistance. The first example I'd like to show you involves glyphosate, which Pat has already covered its mode of action. Glyphosate works so well in part because it translocates really well throughout the plant. On the left, you can see a susceptible sorghum plant or Johnson grass, and the red arrow indicates where a radio-labeled glyphosate was applied. And you can see that after a certain amount of time, the herbicide moves really well throughout the plant. You'll notice the roots have a high concentration, and the younger leaves and the growing points, glyphosate is moved. But on the right, you'll see where glyphosate was applied, but yet much less of the herbicide actually reaches the roots or, in particular, the younger leaves. So by this way, the plant is able to sort of protect the tissue that's at the younger growing points and survive. It may show some damage, but at the use rate in the field, these plants will survive and contribute progeny to the next generation, and you'll see evolution of resistance through this mechanism. Now, how is this actually occurring? Because glyphosate does translocate so well, it is crossing a number of different membranes throughout the plant. It's crossing into the plasma membrane and the symplast when it enters. It potentially needs to cross into the chloroplast, so across the chloroplast membrane to reach its site of action. In some cases, such as sorghum, it's not clear exactly how glyphosate is showing reduced translocation, but it may be changes in some of that membrane transport, either into the symplast or into the chloroplast. But there's a really interesting example that has been documented in horseweed or Canyza canadensis and a couple of other species now. And in this case, the plant is actually able to sequester the glyphosate into the vacuole. So we know the vacuole is a great storage place in the plant cell for anything that could be toxic or no longer needed, because it can be stored in the vacuole and kept away from the rest of the cell. What's been discovered in horseweed is, first of all, in a study in 2010 using P31 NMR, The authors demonstrated that glyphosate could actually move from the cytoplasm at a neutral pH into the acidic pH of the vacuole, and this pH shift causes a shift in the NMR absorbance spectrum. So they could document that glyphosate was moving cellular compartments. And further, this mechanism seems to be temperature sensitive. What you're looking at on the right demonstrates what happens when you apply glyphosate to horseweed in the cold, so at 10 and 8 degrees day-night cycle. These are resistant horseweed plants, but they're controlled by typical use rates of glyphosate, whereas the same population, when you treat it at warmer temperatures, 30 and 20 degrees Celsius, you can see that the plants are resistant. Again, a typical use rate here at 0.84 kilograms per hectare, and they're resistant up to several times that. Combined with the NMR data, the authors were able to show that in the cold temperatures, the glyphosate really wasn't going into the vacuole. So it suggests that this could be an active process that's temperature sensitive. More broadly, we do see this for a number of herbicide resistance mechanisms, that they can be affected by temperature. So the mechanism tends to function well in the environment in which it was selected. So that makes sense from an evolutionary perspective. If the mechanism was selected in a warm environment, it will tend to function best in a warm environment. There are cases where the resistance mechanism was selected in a cooler environment, because that's the environment in which the herbicide is typically used. So you can see here the case of sequestration into the vacuole. This does appear to explain some of the cases of reduced glyphosate translocation. Very recently, this paper was just published a couple of weeks ago. What you're looking at here is an example of reduced translocation for the herbicide 2,4-D. This herbicide has a different mechanism of action. It mimics the plant hormone auxin, and by applying a large dose of auxin to dicot or broadleaf plants, it triggers a chain of events that essentially deregulates cell division and cell growth, causing death to the plant. 2,4-D also, like glyphosate, tends to translocate quite well. In this study you can see they've applied radio-labeled 2,4-D to a leaf in a susceptible plant of wild radish here, and you can see that the 2,4-D has moved really well throughout the plant, to the roots, to the growing points. But notice in the resistant plant, 2,4-D is applied to an older leaf, and it really just stays right there. It is not moving to the roots, it's not moving to the growing point. So in this case, it appears that 2,4-D is not even able to access the phloem for that transport to the rest of the plant. It's quite interesting in this study from Steve Powell's group at the University of Western Australia, that in susceptible they could actually use some physiological inhibitors of plasma membrane transport and reproduce this resistant phenotype in susceptible plants. So by blocking transport across membranes, they could show that glyphosate was staying in the treated leaf. So it remains to be seen exactly how this is working in the resistant population, but certainly a very interesting example of how plants can evolve herbicide resistance through reduced translocation to both 2,4-D and to glyphosate. This is another example in a way of reduced translocation, but yet with another twist. What you're looking at here is giant ragweed, or Ambrosia trifida, and the plant on the left is resistant to glyphosate, although you might not think so by looking at it. This photo is taken 24 hours after treatment, and you'll notice that all of the older leaves on the giant ragweed plant have really curled up. They're showing a lot of cell death, cell damage. And why is this? This is not normally how glyphosate works. On the right, you can see a glyphosate-susceptible giant ragweed 24 hours after treatment. Glyphosate's normally a fairly slow-acting herbicide, and this plant will essentially stop growing, and after a couple of weeks it will be dead. But on the left, you can see this rapid response where the leaves have very quickly started to show this rapid cell death. And in this way, it appears that the translocation to the growing point is reduced. The plant can almost survive by amputating the older leaves that have absorbed the glyphosate and prevent it from moving to the meristem. How exactly is this happening? A number of groups have worked on this. Giant ragweed is found through the upper Midwest in particular and southeastern Canada. So groups at University of Guelph and at Purdue have worked on this, as well as a PhD student at Colorado State, Christopher Van Horn with Phil Westra. On the top here you can see a figure from Christopher in that the resistant plants right here, you're looking at hydrogen peroxide staining after treatment. So within 30 minutes you can see hydrogen peroxide is starting to be generated, and this could explain only in the old leaves why this rapid cell death is happening. But the mechanism behind this remains unclear. But again, a really interesting example of how plants can just have very diverse ways to evolve resistance to herbicides. Next, I'd like to move into what is becoming a very important area of herbicide resistance research, as Pat mentioned, and that's what's known as metabolism-based herbicide resistance. This happens, we know, through increased expression of certain families of genes, such as cytochrome P450 monooxygenases, glucosyltransferases, and glutathione as transferases. Metabolism of xenobiotics generally proceeds through three phases. The first phase can often be oxidation or hydroxylation conducted by a cytochrome P450. The second phase can be a conjugation to a glucose or a glutathione, and the third phase is often then sequestration into the vacuole or the cell wall. We know this because many of the herbicides that are used on crops, the crops are naturally able to survive that herbicide because they have these metabolic mechanisms. They can rapidly break down the herbicide to a non-toxic form and survive, whereas the weeds might do the same thing, but they do it much more slowly normally. So the herbicide works to control the weeds and it doesn't damage the crop. But we see those same mechanisms evolving in weeds or similar metabolism phenotype. The issue here is that it has the potential for unpredictable cross-resistance because these genes are not at the site of action of the herbicide. Therefore, they could potentially have activity on herbicides from different modes of action, and unfortunately on new herbicides that haven't even yet been discovered. If they have some chemical component that's similar to an existing herbicide, these metabolic mechanisms can potentially confer resistance. We think of this as more of a generalist or a broad spectrum adaptive response in comparison to target site resistance that tends to be, you could think of more as a specialist adaptive response. I'd like to highlight an interesting example from Europe. This is data from Roland Beffat with Bayer Crop Science. They've been monitoring target site resistance and metabolic resistance in a number of weed species for quite a few years. What you're looking at here is the weed species blackgrass from southeastern England, and these are all populations that are resistant to the ACCase herbicides. These control grasses by inhibiting lipid synthesis. You'll notice that all the dots are red, so that means that these all have target site resistance, which is specific only to the ACCase herbicides. Only about a quarter of the populations have metabolic resistance. But if you go to France and you have a different situation in a different weed species, Lolium rigidum or ryegrass, again looking at ACCase herbicides, in this case 3/4 of the populations have metabolic resistance and only a quarter have target site resistance. Again, the key thing about enhanced metabolism is that it can confer resistance across different modes of action. For example, some of these populations may have been selected with ACCase herbicides, but they might be resistant to the ALS herbicides, a different mode of action, before ALS herbicides were ever used, because the metabolic mechanism can also metabolize the ALS herbicides. This is an example of the metabolic pathway for diclofop methyl in wheat. This is an ACCase inhibiting herbicide that wheat metabolizes very rapidly. So you can see the different steps here. The critical one is diclofop acid is the active metabolite of the herbicide, and wheat has cytochrome P450 activity that rapidly hydroxylates the diclofop acid to a form that's no longer active, and then glucose conjugation happens that allows then this compound to be sequestered. The reason why metabolic resistance can be more difficult to study than target site resistance is that genes such as cytochrome P450s and glucosyltransferases are gene families. For example, Arabidopsis has more than 250 gene models for cytochrome P450s, perhaps up to 305 different P450s, and as well a diverse range of glucosyltransferases. So it can be very difficult to even pick which cytochrome P450 do you think is metabolizing this herbicide, because there are so many. Their expression can vary as well as their sequence. This gives, of course, rich material for selection and evolution because you have a lot of genetic diversity in function and regulation, particularly in cytochrome P450 genes. This shows you really how this can look in terms of the comparison of metabolism in wheat for the ALS herbicide chlorsulfuron and the ACCase herbicide diclofop. These are studies conducted in Australia for these two herbicides. On the left, you can see chlorsulfuron is selective in wheat because wheat rapidly metabolizes it to this non-toxic form. You're looking at HPLC chromatograms of the radio-labeled herbicide. In susceptible ryegrass, or Lolium rigidum, you can see that most of what is applied stays as active chlorsulfuron, but in a resistant population, much more of it is converted to the non-active metabolite, similar to what's seen in wheat. On the right side, you see a similar situation for diclofop. Wheat rapidly converts diclofop to non-toxic forms. Susceptible ryegrass does generate a little bit of the non-active form, but most of it stays as diclofop, whereas in resistant, you still have some diclofop acid around, but you get a lot more of it converted to the metabolite, enough that the individuals can survive. I'd like to take you through some studies that are starting to make some progress into understanding what P450s and what other genes may actually be involved in the resistance. This starts with a recurrent selection experiment conducted by Neve and Powles on ryegrass. They started with an initially susceptible population of ryegrass and conducted recurrent selection over 3 generations. You can see the initial selections were at less than the recommended rate. The third selection was at twice the use rate. By the end, this population was 40-fold resistant relative to susceptible. You can see this on the right, where you have the highly resistant individuals up to 750 grams of diclofop compared to the Australian field dose of 375 that completely controls susceptible. We know that there are no target site mutations in the ACCase gene, and we know that the resistant selected population shows enhanced diclofop metabolism. But what is the genetic basis of this? It's remained elusive until the development of more recent sequencing technology, such as the Illumina platform, that does enable us to look at the gene sequence across the whole transcriptome of non-model species. So in this experiment, the Illumina platform was used, and a protocol called RNA-seq was used, where you sequence the transcriptome, all of the expressed mRNA, from a number of different individuals, and then compare them for their expression. This way you can actually compare the expression of all the P450s at once and try to identify those which might be upregulated in the resistant population. You can see there are biological replications and then some bioinformatics to align the sequence data to a reference and quantify the expression. This gives you an idea of what it can look like after you do the sequencing and the alignment. This shows an example cytochrome P450 72A gene, and each of these black lines represents one Illumina read that was sequenced in R relative to S. So immediately you can visually see the increased expression of this P450 in R relative to S. It doesn't mean necessarily that this is the P450 causing resistance, but this one can then be a candidate which can go on for subsequent validation and hypothesis testing. And that's what was done in this study to look at co-segregation of some candidate genes in an F2 to try to find those genes that have upregulated expression that would co-segregate with resistance in an F2. And this did identify some potential candidate genes which fit the expected model that we know for diclofop metabolism, including 2 cytochrome P450s and a glucosyltransferase. what this figure on the right shows, it's a principal component, but essentially these are all the susceptible individuals in an F2 compared to all the resistant individuals in an F2. And all of these different genes more or less show upregulated expression in the resistant individuals, in particular these P450s and the glucosyltransferase. What's especially important to point out in this study, although this population was selected with diclofop, which is an ACCase herbicide, it did show cross-resistance to some ALS herbicides that can be metabolized, even though it had never been selected with those ALS herbicides. So again, it shows the challenge of metabolic resistance. Another challenge is highlighted there in that metabolic resistance tends to be a complex genetic trait. That is, it involves more than one gene, potentially two or more, making it more difficult to study the inheritance, more difficult to identify the resistance mechanism, and in some cases more difficult to manage. Another study to point out, this is also in ryegrass but with ALS herbicides. Again, you can see that the study identified increased expression of two cytochrome P450s, a glucosyltransferase, and a GST. In this principal components analysis, you can see all the red dots are resistant to ALS herbicides, and more or less they show increased expression of those four genes. It's perhaps not the whole story, but you can consider these as transcriptional markers. There are, at a minimum, genes that are associated with resistance, even if they're not necessarily causing resistance. This is another really interesting example I wanted to point out today. This is again in blackgrass, or Alopecuris myosauroides. In the UK, there's a population there called Peldon that is resistant to multiple herbicides. It's been studied for quite a while. And in this case, they see upregulation, very strong upregulation of a particular GST from the Phi class. And in this blot, you can see in the multiple resistant blackgrass and in a multiple resistant ryegrass, the increased expression of this GST from 4 to 13-fold higher relative to a wild-type susceptible. And here you can see two different herbicides. The first one is chlortoluron. This inhibits the PS2, so essentially this inhibits photosynthesis and causes death to the plant. So you see a wild type is controlled by chlortoluron, and the resistant is not. But when you add this compound NBDCl, this is a known GST inhibitor, you can see that the plants actually become more sensitive to chlortoluron. Similar, they found for phenoxaprop, which is an ACCase herbicide. Of course, wild type is controlled by phenoxaprop, the resistant is resistant. But when you add the GST inhibitor, it knocks out some of the resistant and the plants become more sensitive. So it really suggests that this increased GST expression has a role in resistance because the inhibitor reverses it. In some cases, the GST may function directly to conjugate herbicides. That seems to clearly be the case for atrazine. But for some of the other herbicides, the GST may have a more complex role in actually regulating the biochemical activity of other proteins. The final metabolism example I'd like to show you is in water hemp, or Amaranthus tuberculatus. This is from the research group at University of Illinois. Mesotrione is part of the HPPD mode of action group of herbicides, the most recently introduced mode of action in the mid-1980s. This also interacts with photosynthesis. And these herbicides are generally selective in crops such as corn because corn can rapidly metabolize the herbicide to an inactive form, whereas the weeds cannot. So here on the left you can see a comparison between resistant and susceptible water hemp, and clearly the susceptible water hemp has a much higher level of mesotrione active, whereas the resistant is converting it to this other metabolite. And then on the right, you see again the usefulness of a physiological inhibitor. In this case, they're using the compounds malathion and tetcyclacis, which are both known to inhibit cytochrome P450 enzymes. So in corn, you can see that after a certain amount of time, only a small amount of radiolabeled herbicide remains as mesotrione. But if you treat it with malathion, then a lot more of it remains as mesotrione, suggesting that a P450 is being inhibited. And you see the same response in resistant. So a lot of the mesotrione is being converted to something else in resistant, much more than S. But when you combine it with malathion, a P450 seems to be inhibited, and you maintain a lot more of the herbicide in the active form as mesotrione. So again, an example of a crop-like mechanism evolving in a weed involving herbicide metabolism. The final point to cover here today is that, again, as Pat mentioned in the introduction, the era of single herbicide resistance has been happening, but really in recent years what we see is that multiple herbicide resistance is increasing. What you're looking at here is the number of weed species with resistance to 3, 4, even all the way up to 11 different modes of action within a species. And we see this trend continuing because different resistance mechanisms can accumulate, particularly in cross-pollinated species. You can see that here. What are the actual weed species that show multiple resistance? Lolium rigidum is one of the worst offenders, of course, with 11 different sites of action resistant. The Echinocloa species are certainly up here, Alipicurus, and the Amaranthus species, which we've spoken about this morning. And one of our local favorites here in Colorado, Kochia, at this point has 5 sites of action of resistance which have been reported. This timeline shows you how resistance has evolved in water hemp over time, and you can see that it is this cumulative process. Single modes of action, the PS2s and ALS, then being reported together, then glyphosate resistance would evolve and then occur together with ALS and PPO to where now you actually have you have glyphosate, PPO, ALS, and HPPD resistance occurring together along with the triazines. So you just get more and more complex multiple resistance patterns and that even can occur within a single population. Here is the number of species for which you have multiple resistance within a single population. So there are a couple species out there for which one population is resistant to 5 different modes of action. It becomes very difficult to manage. This photo from Pat illustrates how water hemp is truly resistant within one population to 4 different modes of action. These other controls are here to demonstrate that the different herbicides are working. And you can see they're not that happy, but they would certainly survive and contribute progeny to the next generation. Kochia is another one in which you have 4 different modes of action confirmed within a single population, including dicamba and glyphosate. And ryegrass from Western Australia has been confirmed in surveys that the vast majority of the populations have multiple resistance, that is to 2 or more herbicide modes of action. In fact, you can see in this snapshot of the Western Australian grain growing region, there are very few populations that are only resistant to 1 herbicide. And the final point is that in a lot of these populations, they found enhanced metabolism, and it often occurs in combination with target site resistance. So it's not just metabolism or target site alone, but that these different mechanisms combine to confer ever higher levels of resistance. A final illustration that we thought everyone would find interesting is that it's not just herbicides, of course. Weeds have been evolving resistance to the control measures imposed by humans for thousands of years. And that includes hand weeding. You're looking at an example here of barnyard grass or Echinocloa from rice systems. And on the left you have rice with a green stem and upright leaves. And on the right you have barnyard grass with a red stem and droopy leaves. So you identify this, you pull it out by hand, and it doesn't set seed. But in the middle is actually barnyard grass with a green stem and upright leaves just like rice, and it's resistant to hand weeding. So no matter what, weeds have a lot of genetic diversity to evolve resistance to the things we try to do in our cropping systems. Certainly we know there are new modes of action coming in the near term. So we know that target site resistance can be managed by rotating to different modes of action, but as we've highlighted today, metabolic resistance is really a big threat because of these cross-resistance patterns to other modes of action, combined with the phenomenon of multiple resistance, combination of different mechanisms. So it's clear weeds have a fascinating range of resistance mechanisms that evolved, and we thank you very much for your attention today, and we would certainly be happy to take any questions.
Thank you very much, Todd. Again, yes, I see several questions that have come in. One is, again, from, let's see, Here we go, from Dan Kunkel. Do you have any indication why the cultivated crops, corn, wheat, soybeans, seem to have higher levels of GST, P450s, et cetera, than weeds do? Or did humans have some role in the selection process, albeit unknowingly?
Well, yeah, that's an interesting question, because we do see that a lot of the crops have high activity of certain P450s. It could be a bit by chance in that we have screened for herbicides that are metabolized by those P450s in crops but not in weeds. So we could find perhaps other P450s that have much higher expression in weeds than in crops, but through the herbicide development process, we're identifying those that have higher expression in crops. It could also be part of the domestication process, or even, you know, part of hybrid vigor, in that perhaps whatever these P450s are doing normally contributes to the crop productivity. I don't know if Pat might like to add anything to that.
Pat, have any comment?
No, I think Todd kind of hit it there, hit the nail on the head. Primarily, it's an artifact of selection. When a company is trying to come up with a herbicide, they're looking for those that don't kill the crop and kill the weed, and so they happen to select those P450s that are important in the crop.
Okay. Here's another question for Todd. Why do there seem to be more cases of metabolic herbicide resistance in grasses than dicots?
This could be due to a number of different factors. It could be partly due to, this is what we've observed so far, in that the ACCase and ALS herbicides were in a lot of cases used on grasses in cereal crops, and because they're selective in the cereal crops through metabolism, that gave us the opportunity to find it. And partly it's because target site resistance is easier to identify so far, because we know what the target site gene is, we can sequence it and find the mutation, and sometimes that's where the research has stopped and it hasn't gone on to look for metabolic resistance. I think now, as metabolic resistance in dicots is starting to be identified, such as the HPPD resistance, as people are looking, there now have been some reports of resistance to triazine herbicides and to ALS herbicides in dicots through metabolism. So it partly might be because we just haven't looked for it very much in dicots, and it could be because there are, you know, a number of grass weeds in cereal crops evolving resistance to selective herbicides for which metabolism is possible.
Okay, here's a question from Sandeep Rana. For either one of you, do you have any comments about the role of epigenetics in conferring non-target site resistance?
I would comment that it could potentially have a role, because we see the change in expression of certain genes, such as P450s. So you could have an epi-allele that could influence that expression. So perhaps a loss of methylation in the promoter could lead to an increased regulation, and then that can be inherited and potentially lead to a heritable increased expression of that gene. And we know that the inheritance of metabolism resistance is generally complex, which could fit with epigenetics in terms of potential reversion of an epi-allele to its former state. But, you know, so far I think we haven't really identified how these genes are upregulated. So it could be through mutations in promoters, mutations in transcription factors, epigenetics. I think we need to consider all possibilities for how genes can be upregulated.
One more question. This is from Bruno Vieira. Is there any case of herbicide resistance due to alternative splicing of the target gene transcript? Do either of you have a comment on that question?
None that I'm aware of.
In a way, the PPO example would be, it's not exactly alternative splicing though, is it? It's an alternative translational start site. So that's actually happening post translation right, for the dual targeting?
Right, and that particular part of that story probably didn't evolve specifically as a resistance mechanism. That's a natural variation of that gene in water hemp as well as other plant species.
Yeah, I'm also not aware of any alternative splicing involved in herbicide resistance, but it could be possible, particularly for maybe some of the metabolic genes. You might have one splice variant that normally is rare, but that's what has activity on the resistant gene, and you could select for increased expression of that particular splice variant. Could be possible.